In this video, we are going to talk about the chemical and molecular basis of muscle contraction. The process of muscle contraction is well explained by a theory called the sliding filament theory. During muscle contraction, the length of the muscle decreases. Think of it like a rope pulling a lever. Muscles are densely packed with specialized proteins and each plays a distinct role. We can categorize the proteins in the muscle into three main groups. First, the contractile proteins. These are the ones directly involved in the actual contraction process. Second, the regulatory proteins, which tightly control the contraction. And third, supportive proteins, which provide structure, elasticity, and hold the contractile and regulatory proteins in their place. Every skeletal muscle contains individual muscle cells called muscle fibers. Muscle fibers are highly specialized cells designed for one main function, the muscle contraction. They contain numerous mitochondria to supply the energy required for contraction and a well-developed sarcoplasmic reticulum that stores the calcium ions. The interior of the muscle cells is filled with long thread-like proteins called myofibrils, which occupy most of the cell's space and push the nuclei towards the periphery. Just like a red blood cell fills its cytoplasm with hemoglobin, a muscle cell fills its sarcoplasm with myofibrils to gain functional excellence. If we zoom in and take a cross-section of a single muscle fiber, we see this intricate pattern. Notice the two main types of filaments visible as dots in this cross-section. The thicker structures shown here in blue are the thick filaments made up of protein myosin. The thinner strands highlighted in yellow are the thin filaments made of actin. The myosin protein form long tails that spiral together like braided hair, while the tiny heads project outward in all the directions. Each thick myosin filament is positioned to interact with 6 surrounding actin filaments. The thin actin filaments are anchored at their ends to a dense wall known as the Z line or the Z disc. The entire structure from one Z line to the next is known as a sarcomere. This repeating unit is the fundamental contracting engine of the muscle. While actin and myosin are the main structural proteins of the sarcomere, other proteins are essential to regulate contraction. These proteins are troponin and tropomyosin. Naturally, an attractive force exists between myosin head and specific points on the actin filament. If these sites are unhindered, the myosin head maintains strong constant attachment with the actin filament, leaving the muscle in a rigid state. This problem is solved by tropomyosin. It spirals around the actin filament, effectively blocking the attractive binding sites. This means tropomyosin positions itself between the actin and myosin, physically hindering their natural attraction. In other words, tropomyosin helps the muscles to relax. When we need to contract, this inhibitory effect must be removed, meaning something must pull the tropomyosin away, unmasking the attractive sites for the myosin heads. This action is handled by the troponin complex. This regulatory protein is a trimer made up of subunits troponin C, T, and I. Troponin C has a binding site for calcium ions, troponin T has a binding site for tropomyosin, and troponin I has a binding site for actin. Troponin regulates the position of tropomyosin. When calcium enters the sarcoplasm, it binds with troponin C, which triggers the troponin complex, bringing a conformational change. This change causes troponin to pull the tropomyosin away, unmasking the myosin binding sites. Myosin is the key contractile player. Its head features an ATP binding site. A binding site is simply a molecular pocket or location on the myosin head that has a natural affinity to recognize and temporarily bind with an ATP molecule. Once bound, the myosin's own ATPase activity rapidly hydrolyzes the ATP. This released energy changes the conformation of the myosin head. The ADP and inorganic phosphate remain attached to the myosin head until the beginning of the power stroke. Imagine a stretched rubber band storing potential energy. The myosin head works in a similar way. It picks up energy from the ATP hydrolysis, converting it into a cocked configuration. To elaborate, just as you use your own energy to stretch a rubber band, the myosin head uses energy from ATP to physically change its shape and become loaded. The energy isn't used for movement yet. It's simply stored, ready, and waiting to pull the actin filament. ATP gives the energy needed to pull actin, but calcium must give the permission. The entire contractile machinery is triggered by a single critical signal, the calcium. As we said, ATP provides the fuel, but the calcium signal is the key that unlocks the mechanism. Calcium is stored in large amounts within the sarcoplasmic reticulum. Here, dedicated calcium pumps work continuously to keep the calcium contained within the compartment. These pumps restore any accidentally leaked calcium back into the sarcoplasmic reticulum to prevent unwanted muscle twitches. To start contraction, a nerve releases acetylcholine. This signal generates an electrical impulse or action potential that travels across the muscle surface. This electrical signal activates specialized receptors present on the sarcolemma called the dihydropyridine receptors. These receptors are physically coupled with the ryanodine channels on the sarcoplasmic reticulum. When dihydropyridine channels detect action potential, they get activated, triggering a pulling mechanism on the ryanodine channels, like pulling a cork from a glass bottle. When activated, these channels open wide and the stored calcium floods into the surrounding muscle fluid. This sudden burst of calcium is the permission we spoke about. It immediately binds to troponin C, causing the entire troponin complex to change shape and pull the tropomyosin shield away. With the active sites now exposed, the cocked myosin heads are free to attach to the actin filament. The moment the sites are unmasked, the cocked myosin head instantly attaches to the actin, forming a cross bridge. This attachment acts like a trigger, releasing the inorganic phosphate molecule. With the phosphate released, the myosin head violently snaps back into its neutral, low energy position, just like the release of a stretched rubber band. This massive forceful rotation pulls the attached actin filament towards the center of the sarcomere. This is known as the power stroke. Following this rotation, an ADP molecule detaches from the myosin head. During this movement, each individual myosin head pulls the actin filament by a tiny distance of about 11 nanometers. While each myosin head works individually with asynchrony with other myosin heads, the cumulative pull of thousands of myosin heads in a sarcomere bring a overall smooth continuous sliding motion of the entire muscle. Crucially, the myosin head remains tightly locked to the actin until the new ATP molecule arrives and binds to it. This binding alone changes the head's shape, which is enough to break the cross bridge. Note that ATP hydrolysis is not needed to break the cross bridge. Once detached, the new ATP molecule is immediately hydrolyzed, recocking the head for the next pull. This entire cycle will repeat rapidly as long as the calcium is present to keep the binding sites exposed. While holding this box in my hand, my muscles are in tonic contracted state. When my brain commands to leave the box, the drop is instantaneous. Have you ever wondered how muscles relax with such a great speed? Muscles can only achieve full relaxation once every last calcium ion have been pumped back into its storage compartment. This crucial energy-intensive task is performed by a dedicated protein structure known as the sarcoplasmic reticular calcium ATPase pump. These pumps operate continuously both during contraction and relaxation. During muscle contraction, the ryanodine receptors release calcium like a flood, so the release outnumber the reuptake. When the nerve signal ceases, this instantly causes the ryanodine receptors to close. This immediately cuts off the supply of calcium flowing into the muscle fluid. With the release channel sealed, these pumps suddenly become unopposed. They rapidly sequester all the remaining calcium from the sarcoplasm. As the calcium concentration in the sarcoplasm quickly drops below a critical threshold, the calcium detaches from the troponin C. This detachment allows the inhibitory protein troponin I to regain its grip and forces tropomyosin to shift back, once again blocking the myosin binding sites on the actin filament. This final act of remasking the active sites instantly reverses the tension, allowing the muscle to achieve full and rapid relaxation.